barking at the computer.
It will just like, you know, really grab attention.
Yeah, exactly.
All right, so we're, oh, that's not, it's connecting. Perfect. Really grab attention. Oh, there we are.
Yeah, exactly.
All right, welcome back to Ask a Scientist, everyone. My name is Abby with the Asheville Museum of Science. And today we are going to be talking with Dr. Jennifer Honeycutt. So Dr. Jen is an assistant professor of psychology and neuroscience at, is it Bowdoin? Bowdoin, thank you. I did not know how to say that. Should have checked earlier. In Maine. And her research focuses on investigating the emotional consequences to the maturing brain during and after experiencing caregiver deprivation. So before her position as a professor, Dr. Jen spent 5 years as a postdoc at the Northeastern University, where her initial findings kind of indicated that early life adversity leads to earlier brain maturation in females compared to males when using a rat model. So this is going to be a really interesting talk. She studies a lot of the physiology and behind the scenes of the brain. So we're just going to pass it off to Dr. Jen, and don't forget to send in questions. We will pass them along.
Awesome. Here, let's see. I'll share my screen. All right. Can you see that? And it's just the presentation? Perfect. All right. So before I get started, so I have kind of a talk for you, some cool data to share along the way. If you have questions, right, feel free to interrupt. And Abby will make sure that I get those. Just to give you a little bit of background about who I am. So I'm just starting. This is my first year teaching at Bowdoin College, which is up in Maine. I have my bachelor's from Colby College, which is also another Maine small liberal arts college. So I'm very happy to be back in Maine and to be teaching at a small liberal arts college that really puts a lot of emphasis on research. So I'm starting up my lab now. As a scientist, I'm a first generation college student. So I'm the first in my family to go to college and to earn a bachelor's and to earn a PhD. I am also a member of the LGBT community. I am married, and my wife and I just had a daughter, and she is 4 months old. So if you hear her, apologies, but now that we're working from home, that is life. Also, you will probably see and or hear my dog come tearing in here at some point because we'll be playing back some rat vocalizations, and every time he hears them, he gets really excited. So if that happens, again, I apologize. I'll try and calm him down, but we'll be doing that. So really what I want to talk to you a little bit about today is what is caregiver deprivation and early life adversity? How does it shape our brain? And then we're going to kind of pivot a little bit to how can we study this even further using ultrasonic vocalizations that rats kind of emit or send out to one another to better study anxiety and emotion in an animal model. So just to kind of orient you on what we're going to be talking about so you can kind of keep track with me, I'll give you some background on early life adversity, what it is, how we can model it, some methods of how I go about looking at this in the lab and how I look in the brain, adversity-induced changes. So what kind of differences do we see when it comes to sex, so males and females, as well as age-dependent effects across development. And we're going to focus in on the neuroanatomy of this. So we're going to look at some brains. And we're also going to talk a little bit about the behavior that's associated with that change. And then we're going to talk about how can we better probe anxiety? How do we get a better picture of what is anxiety and what is driving anxiety in a brain? We're going to talk about a new way of inducing anxiety that I've been working on. As well as how we can use that to identify circuitry in the brain that's important for emotion, which is very, very difficult, right, to study in animals. In humans, we can ask somebody, how are you feeling? What are you feeling today? And in a rat, we have to really just kind of infer based on behavioral data and brain data what that animal might be experiencing. So right now, the tools that we have are good, but they could always be better. And so we're going to talk about how we're trying to make that better so we can better understand it. And then I'll end with some conclusions, right? And then there'll be time for questions, but again, we can stop here and there if questions do come up. That's absolutely fine. So what is early life adversity? So early life adversity or early life stress, these are often used interchangeably. I'll usually refer to this as ELS, short for early life stress. And this is really talking about childhood adversity and maltreatment. So in people, when we think about early life adversity, we're really talking about what has an individual experienced during early development. So as a child, how is the environment, how you're treated, who your caregivers are, and the type of caregiving you're receiving, how does that really affect you as a person and your development? So this really runs a very wide range of things that we're talking about. So childhood adversity can range from abuse, emotional or physical. It can be caregiver deprivation, so being left alone in an environment where you don't have any support, etc. Right? It's a very, very wide range. We know that early life adversity in many forms increases later risk for psychiatric illness. And we think this is largely due to changes in brain development. So when the brain's developing, it makes connections between different brain regions. And if you're experiencing adversity, there's a lot of evidence to suggest that there's disruption in that development of those brain circuits. One that's particularly important is your prefrontal cortex. So this bit of brain that's right here at the front, this helps us to really kind of inhibit perhaps inappropriate things. It helps us to tell other parts of the brain that we don't need to be as excited about certain things. And this is the part of the brain that allows us to kind of make reasoned decisions. And this doesn't fully develop until we're about 23-ish years old. So this is the last part of the brain to develop. And so in this figure that we see here, we see the brain, and across age, we see that it's getting bluer. So that blue is maturation, right? So as we get older, our brain matures, and that maturation is signified by a decrease in the number of cells. All that means, it doesn't mean that the cells are kind of there's something wrong. It just means that the brain is fine-tuning itself. So that way we have a better circuit and it's more efficient. So once that's completed, we have what should be an efficient circuit. What we know from previous studies is that individuals who have experienced early adversity in many forms, and this is in humans, we see that this gray matter, right, these cells in the brain, especially in the prefrontal cortex, they actually show that they're more mature earlier in people with early life adversity history. So we see less gray matter earlier on, so far before we would normally expect that to happen. And we also see that there's an increase in trait anxiety in these individuals. So not only does their brain look like it's maturing too quickly, but we also see an increase in anxiety-like behavior in these people. And so what I really want to focus in on here is usually when we think about becoming mature, this sounds like a good thing, right? When you would want maybe your brain to mature more quickly. But in this particular instance, the brain isn't prepared to mature this quickly. And so if these things are happening and the different parts of the brain are trying to talk to the prefrontal cortex too soon, it's not prepared to integrate all of that information appropriately. So that's where we think we start to run into issues where the brain cannot compensate because it's now a little bit disorganized because things are happening too quickly. So when I talk about this communication, especially when it comes to adversity and anxiety, I'm talking about the cortical limbic system. And to break down what the cortical limbic system is, it's really the kind of the insides of your brain, these emotional centers of your brain that send axons from the cells. So the cells are communicating from this part of the brain to your prefrontal cortex and other parts of the brain as well. But this very specific kind of cortical limbic system, we see changes in the connectivity between these two and the way that they talk to one another in early life adversity. right? And so we see this in humans, and we're able to look at this in humans and these associated changes in how we then perceive possible threat, for instance, or how we become anxious, using the MRI, the magnetic resonance imaging. So if you've ever been in that big MRI, right, that's what we're talking about here. And we can use the fearful face task in humans. So this is what the fearful face test looks like. You're sitting in an MRI, you're presented very quickly with faces with different emotional expressions. And if we look at brain activity while we're doing this, and you're looking at a fearful face, so a face that looks like, ooh, like something's happening, right? We're very visual when it comes to facial communication. And that's how we get a lot of information from our environment and from other people. So if somebody looks nervous, right, you're probably going to look over your shoulder and be like, well, why are we nervous? Right? What's going on here? And so when you see this in the MRI, part of our brain, the amygdala, that is important for alerting us to possible threat is going to become active. And then it's the job of the prefrontal cortex to help integrate information to say, should we be anxious about this? Should we be afraid? Is there things to indicate that there's a threat? If it says no, it'll say, shush, 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 like we're fine, right? Everything can calm down. And so what we see in kids, and so in this graph here, if you see things above the line, that means that the amygdala is driving the conversation between the amygdala and the prefrontal cortex. If it's below the line, the prefrontal cortex is driving the conversation, saying, no, 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 we don't need to be upset about this. In kids, we see an increase in the connectivity between these two. So the activity of these when they're looking at a fearful face. And in adolescence, we see that the prefrontal cortex can say, no, we're good, right? Now that circuit is starting to develop. What's interesting is if we look at kids with a history of institutionalization, so these are kids that have experienced rearing or growing up in an orphanage with severe caregiver deprivation, we see that children look like adolescents. So their brain is developing more quickly, right? So here, just to kind of summarize that, we see these developmental changes in connectivity, then they're accelerated following early life stress. Yep.
I don't know if you're going to touch on this later, but I was just wondering, what is like sort of the criteria that you have in the lab to simulate early childhood stress?
That's next.
Okay, cool.
Yeah, perfect. No, that's perfect. Thank you. So before we get into the nuts and bolts of that, I want to just kind of talk really quickly about how this particular circuit in a rat brain, and then we'll touch on that. So this is what a rat brain looks like. This is a section kind of like how you'd slice bread, so like this. And you can see this black portion here where it says BLA, that's part of the amygdala that becomes active and potential threat. And it sends parts of those neurons called axons to the prefrontal cortex to tell it, hey, there might be something wrong here. And so you can see those kind of fuzzies in the B version. Those are axons that have arrived in the prefrontal cortex, specifically from this part of the amygdala. We know that this takes a long time to develop. And so this is why we think that this is a prime target for why we see this long kind of latency between early adversity and when somebody might have an anxiety disorder or depression, because the circuit takes so long to fully develop. And so you can see right down here, we have P6 through P65, P meaning postnatal day, or the number of days old this rat is. And as the rats get older, you see more of these axons have arrived over development. All of this research has been limited to males until now, especially this anatomical research, which is a pervasive issue in behavioral neuroscience, that we're frequently doing in these preclinical models, and we're only looking at male animals, which is a huge issue. So really we wanted to add females into the mix because ELS preferentially and overwhelmingly impacts female connectivity. And females are more likely to develop anxiety and depression after early life stress. So the nuts and bolts of how we actually model this in an animal. So when we're talking about early adversity, and in the model that I use, we're talking about caregiver deprivation or maternal separation in a rat. So here is a picture of children in an orphanage. This is something that was seen quite a bit, particularly in Eastern Europe. So this is, in this particular picture, the children are separated from one another. There's no caregiver in sight. And so there's very limited contact with any other people during this really formative time in their development, right? In rodents, we can mimic this very closely using maternal separation. So maternal separation we do from postnatal day, so day 2 after they're born through day 21, which is when they move out of mom's cage, right? So that's when they're kind of old enough to get their own place. So that would be kind of late adolescence, very, very early adulthood in humans. And we do 4 hours a day. We take them away from mom and their litter mates. And that's what this looks like. So we have these pups. So these pups are probably around 7 days old. Their eyes are still closed. They're being thermoregulated. So underneath these cups is a water bath that keeps it nice and warm. But you can see the really striking resemblance between how the children are separated from one another and how the pups are separated from one another. So we do this specifically to mimic this kind of very isolated sort of rearing early on. We know from prior research that this leads to a lot of long-lasting consequences. So immunological consequences, behavioral consequences on anxiety and depression, neuroanatomical consequences on how the brain develops, as well as biochemical. And in addition to all of these kind of more neurocentric consequences, we also see health consequences from early life adversity, particularly in humans. Okay. In an animal that's experienced this type of early life adversity, we see from other groups that they have decrease in social investigation. So now we're looking and we can see that the animals are less sociable after, and this is in both males and females. And time again, in the lab that I was a postdoc in, we've seen that animals will spend less time in the center of an open field test, which indicates that they have more anxiety-like behaviors after early life stress. But what's interesting about that is that males don't show this until late adolescence, early adulthood, but females show this in juvenility or adolescence much earlier. So you can see P25 and P45, females show more anxiety-like behaviors, and the males don't show that until a bit later, which is interesting because this kind of mimics what we see in the human literature as well. So the way that I went about determining how this specific circuit develops is we had our animals in-house, were born. We did the maternal separation paradigm. We weaned them, meaning they got to move out of mom's cage and they got their own cage mate. And so now they're kind of on their own. And then I did an intracranial surgery on them to introduce a compound called BDA, or biotinylated dextrin amine. And so what this does is we can inject it into the brain. It stays where you've injected it, and the neurons kind of suck it up. And any of the neurons near that site that we injected it, that take it up, express that throughout the neuron and the axon. So we can actually visualize where the axon is. And so we did this. I did a surgery. We injected 200 nanoliters. You can barely see this amount. It's tiny, 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 into the BLA, the basolateral amygdala. So that's what that looks like if you take the brains out later and you stain them. So you can actually visualize that's the amygdala. And you can see that dark brown, that's all those cell bodies. And you'll see some pictures of what all those axons look like in a minute. We waited a week. We did behavior to look at anxiety, and then we looked at the brains. And we did this at 3 developmental time points. So we did all of that. And then just to kind of reorient you as well, these different ages. So P21 is like a juvenile in human speak. P31 is early adolescence, and P41 is late adolescence, early adulthood. All right, so what do we find? So here, let's see, this slide sometimes takes a while to load because these pictures are giant. So this is looking at the axons that have come from the amygdala into the prefrontal cortex in these animals that either experienced early life adversity through maternal separation or control rearing. So we looked in the prefrontal cortex, and we looked in 4 different regions of the prefrontal cortex. And specifically, we're just going to talk about it in terms of these 2 chunks, the prelimbic and the infralimbic. So these are receiving limbic projections or limbic axons from areas in the limbic system, like the amygdala. So that prelimbic area and that infralimbic area, if we look at those, here we see the male and the female data. So these are pictures taken from experimental animals. And what you'll see, all these fuzzies are all of the axon terminals. So these are the axons that have arrived there, and they're trying to talk to the prefrontal cortex. And the females, and I don't know if you probably can't see my, can you see my mouse? Probably not.
I don't think so.
Okay, so that's fine. So if you look in the P28 and the control and ELS, the females you'll see already have a pretty dark amount of, and there's a lot of those little kind of fuzzies, right? So they have a lot more axons earlier, and we don't see that in the male ELS animals. And so it actually looks pretty comparable to our kind of young adult animals. So it's arriving earlier in these animals. And we see the same thing in the infralimbic. So within the prefrontal cortex part, that kind of part of your brain that's the last to develop, we're seeing that more of these axons are there. So there's a lot more excitement coming from the amygdala into this area, possibly saying, depending on the situation, we should be anxious about something.
And we did have one question come in from Molly. She kind of asked it right as you were going over this. So she was wondering, what are the biomarkers or the markers of anxiety that you look for? So it is going to be the number of cells or axons.
Yep. So in this particular case, we're looking at the number of axons with the thought or the hypothesis that if we have more axons, we might see also more anxiety. And so here you can see this is a close-up version of what those axons look like when we're counting them. And we actually use this program called Spaceballs that puts these little orbs over randomly selected parts of the brain region, and we count how many of those axons are crossing, and then a mathematical model gives us an estimate of how the overall length of the amount of axons that are in that region. So that's what we see here. And then we looked at behavior of these animals in the elevated plus maze. And the elevated plus maze, so here we have a rat, we have 2 open arms, which are kind of anxiety inducing, right? If you're a rat, you don't really want to be in this kind of scary space where you have no protection. And you have these closed arms that are going to feel a little bit more protected. So if you were more anxious, we would guess that you're going to spend more time in the closed arms than you would in the open arms, right? And that's pretty much what we see. So if we look at time spent in the open arms in males and females, control and ELS, we didn't see, surprisingly, anything significant in the males across the different age groups, but you do see a little bit of a dip in the ELS animals. But in our females, by the time they got to 38 days of age, we saw a significant decrease in the amount of time spent in the open arms, which indicates that they're experiencing perhaps higher rates of anxiety. If we look at and I want to just kind of draw your attention to two of these graphs here. If we look at the females at PD28 and we correlate it with the amount of axons that we saw, so we're now looking at, based on the number of axons, can that predict your behavior? We see that it is able to predict behavior in young females. So in juvenile females, the more time they spent in the open arms, or the less of that innervation, right? And conversely, so if they had a lot of axons in the prefrontal cortex, they spent less time in the open arms and they looked more anxious, right? And in males, we don't see that until later. So this mimics what we've seen previously, where females seem to be a little bit more impacted early on. And it's possible that this is being driven in some way by the amount of axons that have arrived in the prefrontal cortex. All right. So just to kind of summarize what we found in this particular instance, we see that ELS drives this kind of early innervation of these axons. So the axons actually kind of growing in and arriving in the prefrontal cortex. And this seems to be sex specific and also drives sex specific anxiety-like behavior. And specifically within that, we see that juvenile PFC innervation, or the number of axons in ELS, is comparable to what we see in the adults. So that early maturation seems to be occurring in the circuit. And that male maturation occurs a little bit later, which again is something that we kind of expected, and that was kind of conserved. This gives us a little bit of insight into the underlying neural basis of why adversity seems to confer or seems to lead to vulnerability to mental illness, particularly anxiety in this particular case. Okay, so increased innervation or increased number of axons from the amygdala into the prefrontal cortex led to anxiety-like behavior in this case. All right, so before we switch gears, any questions?
Yeah, we do have one that is wondering why innervation is going to lead to more anxiety. But
right,
we're about to cover that.
Yeah, so but I can kind of I'll touch on that a little bit. So the innervation or the axon. So anytime something happens in the brain, you have excitation of neurons. And so the end point of that excitation is neurotransmitter release from the ends of axons. And so if a part of the brain becomes active, so for instance, if we see a fearful face or something that could be a threat, our amygdala is going to become active. That's going to send excitatory or signals that are going to talk to the prefrontal cortex and say, hey, we think something might be wrong. And the prefrontal cortex can then act accordingly. If we have more of those amygdala axons or innervation into the prefrontal cortex, presumably we have more of that signal, right? So that signal is going to be amplified, right? So that's why we're interested in looking at that, because it's not supposed to be screaming, right, to your prefrontal cortex, there's something wrong. It should just be like, I think there's something wrong. But in this case, we're really amplifying it.
Yeah, so we want to be poking it. We don't want to.
Yeah, we just want to poke, and we want to be like, we're good, as opposed to, this is something and we need to pay attention right now, right? So, and we think that might be contributing to kind of hypervigilance in some ways in individuals that do have anxiety disorders or are more at risk for kind of anxiety. So now we've kind of talked a little bit about which brain regions and circuits contribute to anxiety. This cortical limbic circuit is really important, but I'm also very curious about how we can better investigate this and how can we ask this question in a way that's more translational. So how can we study in an animal emotion, a very subjective sort of thing, and make it more objective? So when we study anxiety circuitry, mostly we hear about the amygdala, right? And this emotional area of the brain. And we know that cortical limbic circuitry is really important for both fear and anxiety related behaviors. But there's another region called the extended amygdala. More specifically, it's called the bed nucleus of the stria terminalis, or the BNST, that we found to be incredibly important in maintaining anxiety-like states. So once the amygdala is saying, hey, oh, there might be a threat, now it's going to also talk to the BNST, and the BNST may or may not maintain that activity, and then that could lead to a state of anxiety. So here's a rat brain. So we're looking at the side of the rat brain, and we can see the prefrontal cortex, that blue area. We see the BLA, which is what I was studying before, and the connections between the two. And then we also have the central amygdala, which is important for fear. And that's going to become active, and it's talked to from the basolateral amygdala, and it's going to talk to the hypothalamus, which is going to increase adrenaline, so that fight or flight response, as well as brainstem regions, which could lead to either active, running away, or passive, freezing, fear responses. So the central amygdala is going to be kind of our node for fear when we have BLA activation. But the BLA is also talking to the BNST. And when it sends information to the BNST, it's going to increase HPA axis or activity, so hypothalamic pituitary adrenal axis. It's also going to talk to the brainstem and initiate autonomic responses. And we also know that this is highly sexually dimorphic. It's one of the more sexually dimorphic, meaning that it's different in the sexes. So males versus females, we see differences in how the BNST behaves. So this is a really interesting area to think about when we think about, well, what's going on in anxiety and whether or not this might be meeting some of those sex-specific behavior differences. Yep.
So this is sounding a little bit, as Molly points out, that anxiety has a strong genetic basis. So is this true for mammals, or does environment play a stronger role to anxiety? What do you think?
I think both. I think that kind of goes to the nature versus nurture of anxiety disorders and disorders in general. And I'm a strong believer that you really can't have one without the other, and that these are reciprocal, that any sort of environmental change is going to play on the brain. The way that your brain is through genetics and inheritance is going to dictate how you interact with your environment. So I think it's a very reciprocal sort of system, but suffice it to say, we do see a lot of evidence for heritability for anxiety traits, as well as a lot of influence of environment on anxiety expression. Yeah. So this area of the brain is very interesting to me. And it became even more interesting to me as I started studying and trying to probe anxiety circuitry a little bit more specifically. So I'm interested in my lab in how we can study changes in brain activity and hypervigilance. I'm very interested in this hypervigilance, right? This being very attuned to possible threat as they relate to anxiety, particularly after adversity. So how does adversity maybe make us a little bit more anxious or more prone to this? Yep.
And so this might be something that is, an active field of research, but is there a chance of less susceptibility to mental illness if the adversity is experienced after the maturation on that normal timeline where you would expect it to happen?
Yep. Yeah, I would say it would depend on what we're talking about for mental illness, and whether or not you might have a predisposition that you don't know about. So you could have a genetic predisposition that could be acted on by a stressor later in life, and you could have had a perfectly fine early life experience, or you could have an early life experience that could precipitate some sort of vulnerability. On the flip side, we also see some evidence for resilience after early adversity, where early adversity almost acts as an inoculation to later stressors, and you might be better at bouncing back from those. So that's a great question. So we already talked a little bit about the fearful face task. This is great because we can study these different circuitry, the circuit activity in the fMRI in people by showing people these faces. Sadly, in rats, we cannot do this. We really lack a similar methodology, because if we were to put a rat in an MRI and tried to give them a fearful face task, right, this would not be a very effective task. They would not be able to really tell us much of anything. Their eyesight, first of all, is not awesome. So even though there are very slight changes in rat facial expressions, depending on mood, we wouldn't really be able to pick that up from a rat in a dark MRI, right? We're not going to be able to get much information from that. So I was really focused in my postdoc on, well, how can we make a model or an assay of anxiety that's comparable to what we do in people, but we can do it in animals? So our answer to this was ultrasonic vocalizations, right? So rats emit ultrasonic vocalizations, or USVs, that can convey information about emotional or affective states. And it can also induce cognitive bias for those affective states. And there's two main categories of USVs in rats. We have 55 kilohertz. These are appetitive. So if you tickle a rat and the rat likes you, and it knows you very well and you tickle it, right? Not like stranger to a rat, but like a rat, if he knows you, it's okay. And you tickle the rat, they'll make these giggles, these 55 kilohertz giggles. If you give them a ton of sugar, they'll do this. If you let them play, they'll do this. Versus 22 kilohertz ultrasonic vocalizations, which are primarily alarm calls to let other rats in their colony know something's wrong, or I'm in pain, or something bad is happening. There was a predator nearby. Those are going to be very specific. And so they're aversive because we believe as well that they might indicate that the animal is experiencing a negative emotional state versus the appetitive, where we think that the animal is experiencing a positive emotional state. And so what these look like, so these are spectrographs of ultrasonic vocalizations. These are the 55 kilohertz on the left, which are our happy chirps. And then on the right is our 22 kilohertz vocalizations. And this is when the dog might come running downstairs because I'm going to play these. So we can't normally hear them, but these are downsampled, so they're at our level. And so I just want you to think about, as you listen to them, how do they make you feel emotionally? Do they have an emotional charge to them? Let's see, so that's one of them, and there's the middle one. Right, so they kind of sound pretty cheerful, right? Kind of cute. Now we go to the aversive. So there's 3 trains, I'll let them play. So thankfully my wife shut the door upstairs so the dog didn't come running down. Thank you. So when you just listen to those, though, I mean, the first word that kind of comes to my mind is like forlorn when you hear that last one. It's just very kind of, it really pulls at your heart, and you're just kind of like, you poor thing, right? This animal is in a negative emotional state. We can infer based on the fact that we've been able to categorize the emission or how they're outputting these with what they're currently doing, and differences in brain activity, as well as differences in behavior. Yep.
Do you think, so can the rats hear each other while they're making these noises? Do they have like groupthink? Like, does one affect the others?
That's great. So there is some evidence for what we call emotional contagion. So if they're hearing, right, if there's a rat in the colony that's really stressed out and it's emitting these 22 kilohertz, it's possible that it's affecting the other animals where they're kind of getting a little nervous. On the flip side, if you have the happy ones, they might be like, "Woo, where's their, what's going on? What fun is happening?" So there's definitely something to be said about that. There's some really cool, and if you look at NPR has done a couple of really good like short videos about empathy in rats, where if you have a rat that is restrained and it's emitting these vocalizations, another rat will try and free it. And so there's a lot of really, really cool data coming out where we're looking at this emotionality and sort of metacognition in animals, which is really exciting. All right, so I don't accidentally play this again. There we go. So what I'm interested in is, how can I use what we know about the emotional valence of ultrasonic vocalizations to kind of poke at the circuitry that I'm interested in, the circuitry that I think is involved in anxiety as opposed to fear, right? So I'm not interested in the fear part of things. I don't want to give them predator odor and have them be scared for their lives. I want to know if there's something ambiguous, how are they going to interpret that? And if it continues, is that going to change their state, their emotional state, or is it going to make them look more anxious? And what parts of the brain are mediating that? So in order to do this and to really get a good idea of how we could overlap this with what we see in humans with the fearful face task, I developed these ultrasonic vocalization, or these ultrasonic earbuds that go into an fMRI. And so we can actually put a rat in an fMRI. So here's a picture of our Brooker in my collaborator's lab. And this is the body tube that we put the animals in. So they're awake. They have these little earbuds in their ears, and I can play back 55 kilohertz or 22 kilohertz vocalizations to them while they're in the magnet. And then I can monitor their brain activity while they're listening to this compared to baseline or when there's nothing playing. So that's what I've been doing the past couple of years. And we see some really cool data when it comes to this. And anytime you can see kind of rat ultrasonic vocalization data mapped onto a rat brain from MRI, it's very exciting. So we had our 55 kilohertz playback, we had our 22 kilohertz playback, and then here's a bunch of brains, right? So this is looking through the entire brain. So we have these like bread slices, these coronal sections. From the top right, just as you would read, is the prefrontal area. And then all the way back is the cerebellum. So we're kind of going through the rat brain. And in our 55 kilohertz versus our 22 kilohertz, this is looking at how much blood oxygenation level changes there are from baseline when nothing's playing to when the playback is happening. So we're looking at a difference. And anywhere where you see coloration on the brain is where there was a significant difference from baseline. And this is a lot of really complicated data. And if you're not familiar with a rat brain, this is going to look just like a whole bunch of mess to you. But I want to draw your attention to a couple of spots. So where you see the hot pink and the blue right here, that's the basolateral amygdala. It's only changed from baseline in the animals hearing the 22 kilohertz playback. It's not changing in the animals that hear the giggles, right? So the amygdala is saying, hey, something's going on, there's possible threat. I also want to draw your attention to this part. So where you see, so this is, because I can't point to it, the bottom half of the brain, where you see kind of like those little like eyebrow things down at the bottom, that's the anterior commissure. And then around that we have changes, and that's actually the BNST. So it's a very small region, but we see changes in both the 22 and the 55. And so I'm curious, why do we see differences in both of these playbacks in this area? And I think part of the answer to that is that the BNST has a lot of different parts, and they're differently sensitive to different kinds of stimuli, right? So I'm thinking that they're probably processing information depending on the type of information coming in, and that one part of the BNST might be more tuned into possible threat, and may therefore be more involved in regulating anxiety than other parts. And so in order to kind of figure that out, I did playbacks of this in animals while they were doing behavior, and then I looked at the brains, and we looked at CFOS. It's an immediate early gene, meaning that when a cell is active, within 2 hours, you see an expression of a protein that tells us that that cell was active, and we can stain for it. And then we can visualize which cells were active during the playback period. So if we looked at the auditory cortex, we see that in the 55 and the 22 compared to silence, there's more. Awesome, they heard it, right? So that's a good thing. That's our control. We know that they're all hearing something when we do the playback. If we look at the BLA, we see that there is a significant increase in the number of cells that are active in the 22 kilohertz, and no change from 55 kilohertz from silence. And then if we look at the BNST and we look at two different nuclei, two different subregions, in the oval, we see that it seems to be more sensitive to 55 kilohertz, or these happy calls, where we see more activity in that particular brain region. On the other hand, in the 22 kilohertz, in the anterior dorsal part of the BNST, that's where we see a lot more activity. So I'm interested now in future studies to kind of really hone in on what's happening in this region. And is it particularly sensitive to early adversity, right? So in the animals that I did this in, these were typical animals. A lot of this was pilot work to see, can we see any changes? And the next step is really to say, well, what happens if there's early adversity thrown in the mix? Are they more susceptible to this kind of playback? If we do behavior, so this is elevated zero maze. This is really similar to the elevated plus maze I showed you earlier. Open areas versus closed areas. The only difference is it's just in a track. And if we play back the ultrasonic vocalizations while they're on this track, we see that the 22 kilohertz spend less time in the open areas, which makes us think that it's making them a little bit more anxious, and that this gets exacerbated, or it gets worse, and they look even more anxious the longer that it's playing without any sort of ability to see that there's not a real threat out there. Right, so that's what we see in that. So to conclude the data that I showed you today, We see that early life adversity rewires the brain's development. And this is sex dependent. So if you're a female, it matures your brain a little bit more quickly and looks like it puts you at a little bit of a higher risk to developing anxiety-like behaviors. And this is specific between the BLA and the PFC. I'm now curious, is this also impacted in the BNST? Is the BNST also kind of maturing too quickly, leading to changes in anxiety? Ongoing research is always necessary, but in this case, it's particularly necessary to identify targets for intervention. So what parts of the brain and what kind of processes are happening or disrupted so we can target these better and maybe try and help humans that have experienced adversity to lessen symptoms. And I'm really excited about this because I've been using these ethologically relevant, these really translational social cues to mimic the kind of communication that we would do in humans, to provide this really kind of exciting way of determining and assaying susceptibility to anxiety provoking stimuli. So that way we can try and better understand the neural basis of adversity related pathology in an animal model. So thanks are always in order. My postdoctoral lab, the developmental neuropsychobiology lab at Northeastern University, particularly my postdoctoral mentor, Dr. Heather Brenhouse, who I was her first postdoc, and we did a lot of this really kind of cool work together. And I'm very thankful for her mentorship on all of that. Our graduate students, as well as our undergrads that helped with collect a lot of the data that I showed you today. Our collaborators at the Center for Translational Neuroimaging, Dr. Craig Ferris and Dr. Praveen Kulkarni, the money that makes this happen, because of course, research can't happen without funding, especially government funding. So call your senators, get more funding for research. So we got research from the NIH, and I had research funding from the Brain and Behavior Research Foundation, which funded the completion of those headphones. And of course, thank you to the Asheville Museum of Science and Abby for inviting me. This is really exciting. This is my first talk at a museum and in the Carolinas. So thank you so much for having me. And any questions you have about what I talked about today, about my research trajectory, jobs in academia, whatever you think of, right? I'm totally open to those. So
well, that was fantastic. Thank you so much for coming. I do have a question real quick. So as you mentioned, when you first started out the entire presentation, the brain is obviously super complex. And so it's a multifaceted problem. How do you think events like the pandemic, where we're all staying home, are kind of contributing or simulating that same sort of adversity, either with loneliness, not being in school, or sorts of things like that?
Yeah, I think this is a really interesting time. And I think I've noticed from my friends and colleagues that do similar work, but they do it in human populations. I mean, I've seen so many surveys come out on, for parents of children, like, what is your child doing? How are you dealing with childcare? What does this look like? With the intention to kind of follow these kids and see what's happening. I know that we are in a study, a remote study, that was looking at people who were pregnant during the pandemic and now have had babies, and like what is our mental health? What is, and then they'll follow up at different time points about developmental trajectories, I would imagine. So there's definitely something to be said, because children are kind of being put, in front of screens kind of on their own because there's just too much to juggle. It's certainly not to the same extent of the early adversity that we study, right, where it's extreme deprivation, but the shakeup of things is definitely contributing, I think, to differences in mental health outcomes that we don't even know yet what that's going to look like. But that social deprivation is especially at critical time points in development can be very damaging. So that's something to consider for sure.
And what about something like a more physical issue, like if you're sick or your immune system is very low or you have a traumatic brain injury of some sort? Do you have any ideas of how the same sorts of adversity could affect individuals with those types of predispositions.
Yeah, definitely. I think that anytime you have an accumulation of traumas, that's never necessarily going to be good. You're going to have, it's going to accumulate, and you might have exacerbation, right, of different symptoms. You could, you know, especially if you've already experienced early life adversity, you are already at risk for perhaps being more susceptible to negative outcomes from a secondary trauma. So the prognosis probably, if you have all of those things together, is not going to be as promising as if you didn't have that particular background. However, right, you do have a subpopulation of individuals who come out of something like that more resilient. And something else to think about, and I've heard a lot more lately, and is this idea too that what is anxiety, and this thought that anxiety, because it comes out especially in like college age students, right? It's really protracted. Perhaps one of the reasons that people have pathological anxiety is because they grew up in a specific set of circumstances, and you adjust your behaviors and your brain adjusts to be in that circumstance or to be in that environment. Then once you're taken out of that environment, those behaviors that you've accumulated early in life are now maladaptive because you're no longer in that environment, right? And that could be in some ways precipitating this idea of anxiety, because now you have to completely adjust your schemas and the way that you do things to be in a different space. And I've also heard a lot of kind of anecdotal things where people with anxiety disorders right now, or at least at the beginning of the pandemic, were kind of like, okay, well, I'm always nervous. And now I'm like, now there's a reason to be nervous. So now I'm not as upset that I'm nervous.
Yeah.
Right. So there's something to be said about that too, because now you're put into a space where you're like, okay, well, being anxious makes sense in this circumstance. And so it's almost as though it still impacts you, but maybe not as much as somebody who's never experienced adversity and is now in this adverse situation. And now they're experiencing anxiety for maybe the first time in their lives because they're in this uncontrollable situation.
Right. And I did miss one question that came in the chat that said, how do your findings relate to the pleasure pain balance?
That is a great question. I'm not sure. So when it comes to that kind of pleasure and pain, I think there's a lot to kind of unpack there, especially with understanding in an animal, like where are your pleasure and pain thresholds. I do not do pain work specifically, but I do know that some of the pain work and some other work shows some interesting stuff with emission of USVs. And in some cases, you might have kind of these weird overlaps where occasionally in a situation that might be painful, you might see something that comes close to a 55 kilohertz vocalization, which is, why might we have that? And I don't think we're sure what that is, but that's definitely, a great question and something that I've been thinking about too. And I'm not sure off the top of my head any really kind of recent data that would tell us a little bit more about that.
And so mice and rats are used all the time as sort of our favorite model organism, especially for neuro studies. What would you say we can take away from this study and kind of translate into like the human model, or I guess to walk away with, that's going to help us improve mental health care?
Yeah, I think I think there's a couple of different things we could think about. So from the first bit of data that I showed you, I think from the rat studies, really, this is the only way that we can get a really concrete anatomical answer in a systematic way to really say, oh, X is related to this outcome of the axons, right? Because we can so tightly control everything else in the lives of our animals that we cannot do in people. Even if we do postmortem tissue and we try and look at these axons, you have to consider everything else that person's ever experienced in their life. And we know that the brain is highly plastic, meaning that it's always remodeling itself and always changing and really fluid. And so it's almost impossible to answer some of these mechanistic questions in people. We need these animal models. So if I can stress anything from that part of it, it's that we rely so heavily on animal models and the data that comes from that to understand human disorders, that without this research, we would never be able to identify possible mechanisms. So it's incredibly important. I really hope that comes through. And in the same vein with the ultrasonic vocalization, I think a lot of people look at rats and mice and they think, well, like, what are they going to tell us about humans? Right. And I'm here to argue so much, right? They could tell us so much about us. And we're still figuring out every day. We're like, oh, this is going on in rats too. And we had no idea that they had this kind of complex social structure. We had no idea that they were telling each other kind of an emotional state based on communication. And it makes sense that some of these things would be ethologically conserved through evolution, right? We have all of these really complex social structures. So do rats. They're able to really allow us to answer complex social questions about anxiety, about how our emotions can impact one another in a very compact model system that we can also look at development through, that we can't do in people. So it's a really great way to kind of ask those questions of what is emotion and what drives emotion in a model that we can really kind of systematically investigate.
Awesome. That was a good answer. I like that. And lastly, this is just kind of for me. Do you ever retire any of your mice? Just like, oh, they'll live off in a good place. And would you ever play music for them with your little headphones?
With their little headphones. So they, unfortunately, because I'm a behavioral neuroscientist, I am interested in the brain. So they sadly don't retire. But their brains do. And we get to look and we do beautiful neuroimmunohistochemistry and we can visualize what's going on. And so they're going to a much bigger cause. And so there's something to be said about that. As for the headphones, I'm actually not sure if they can play back music at our range. The transducers that we put in them are ultrasonic. And so I think it would probably be pretty garbled, although now I'm really curious if I were to do any playback, how that would look.
I think of Dolly Parton.
I don't know. I mean, they probably, if it's a good bop, like they'll probably be okay.
9 to 5, they'll be into it.
Yeah.
All right. Well, thank you so much for joining us this morning. It's been a pleasure to talk to you.
Thank you so much for having me. I really enjoyed this.
If there are any follow-up questions, I will just send them to you over an email and we can get them answered for our viewers. And everyone who is watching, thank you so much for your time. I will hope to see everyone back next week.
Awesome. Bye. Thank you so much.